right. Hi, everybody. So we are now here for chapter 7. And this actually is sort of split into 2 parts over the course of the syllabus. But they're all within the same exam. It was just kind of to separate them, just to sort of reorganize their presentation a little bit. So this will be split into 2. And here we go. This is part 1. All right, so sound, this whole, this chapter is, we're basically heading into the other senses. We've covered vision. And so now we're going to be delving into the auditory elements of hearing and listening and how that is processed in the brain, and sort of how we then take what we hear and develop that into language. So we also cover some language production. So this kind of begins with just a basic principle of sound, and that this is the environmental sort of stimuli that we're hoping to connect with in terms of get information from. So we have to transduce, this sort of environmental information to, a neural representation within our body. So looking at the sound wave, sound is produced through compression and refraction of substances like air, metal, and water. The human range of hearing is between 50 hertz, which is a low pitch, and 2000 hertz, which is a high pitch. And again, that's also a very relatively restricted range of hearing in terms of the broader spectrum of frequencies of pitches. The ability to perceive high ranges decreases as we age. I'm going to be looking forward to that, I think. So here is again this sort of like little elements of particles, but that you can see here, this is sort of represented in this wave form. And this is, the ear will be detecting aspects of this wave form. So how does this work? Well, the sound enters the auditory canal right here. And when it travels down, it hits this tympanic membrane. And this is your eardrum. And this is what you can have it infected. And you can see why when you have an ear infection, it can also just be really quite painful. So the tympanic membrane is extremely sensitive. And when air hits it, it causes the action of the 3 bones in the middle ear called the auditory ossicles, which are right here. So you can see these 3 connected to the tympanic membrane. So it basically, affects these and causes them to shift and move, the malleus incus and the stapes. And then they're moving against what is this much smaller, oval window that is right in here. So then you're taking the sound from sort of this larger tympanic membrane and then also narrowing it down to a much smaller window to sort of compress the sound. Now, this large tympanic membrane has the pressure concentrated to push the oval window, as I just explained. And this is because of impedance matching, where this sort of this larger into this smaller, that's sort of this compression element. Right. So hold on, I'm taking a step back here. So once you hit the oval window right here, then this is basically into your full inner ear area, where you've got your cochlea. This is your cochlear nerve. So this is your heading out to your auditory nerve. And then you have your semicircular canals, which are filled with fluid. These are used for balance. And this would then be parts of your vestibular nerve. So you have both hearing and balance in the same information. And this is your euchination 2, where you can, get fluid in your ears and they'd have to clean that out. All righty. So now all the sound has been compressed into this oval window that connects with the cochlea. So what does the cochlea do? The cochlea is basically, you can see here, so this is the whole cochlea, and it winds around. And so this is basically a section of what the inside of this sort of, wound around sort of snail shape. And this is what the inside looks like right here. And here you can see the tympanic membrane. You see how big it is, and here you've got the oval window base sort of just right here. And so what this does, this is actually the whole spread out section of the cochlea. And this is just the same thing sort of spread out, basically wrapped around right here. And here you just see a midsection of that. And so this is area right here would be kind of represented along this frequency space right here. All right, so our cochlea, we have these three chambers separated by these three membranes. They are filled with fluid, and the sound waves then, by vibrating the tympanic membrane and hitting the oval window, which is basically like, sort of, the oval window inside there's not air, there's not space, so when you vibrate, that it's filled with fluid, then it turns the sound waves into these fluid waves. And that's really important for the sound compression then, because it takes, more to travel, for these vibrations to travel through fluid than through air. And so this gets compressed down, and all these converted to these fluid waves that would basically be processed along the cochlea according to their frequency. So this here is what the cochlea looks like. This middle canal here is the organ of corti right here. So this is the really sort of important piece for our receptors in terms of sound. And this sort of just walking through some of these inner pieces. So this is the middle ear canal called the organ of corti. And this again is this oval window, this cochlea right here spread all the way out. And what you can see is that, just as it's not as visible here, but the size of this starts larger, and then obviously the canal gets sort of smaller and smaller and smaller. So this will correspond, this maps on to frequency. So these beginning areas, detect really high frequencies, then medium, and then the more you get towards the back, then it's the lower frequencies. And this may be likely why we lose some of the low frequencies back here when we hear this is some of the stuff that's the first to go. So, and this, if you have to look closely, but this is basically the tectoral membrane right here, which you can kind of see in some of these spaces. And then there's the basilar membrane. And so you can see all the pieces on here. It's just harder to kind of see the layers because they're quite small. So what happens in this organ of corti, or corti, where these receptors are living, because the tectoral membrane is right here. This is then sort of parts of what is being vibrated in here. And then this is the basilar membrane, where you have some of the receptors sitting. So taking a closer look, this is the organ of corti. And now, just to kind of give you a sense and zoom in, this is what you're seeing right here. So sort of hold this sort of spot in your mind, thinking of even this sort of the shape and stuff like that. But this is what we're seeing on the next page. So this whole space, here you have the tectorial membrane, and here's you have the, basilar membrane is this is a basilar fiber. And these right here are called hair cells, and these are the receptors. So let's walk through this. So the basilar membrane right here is a flexible base, and it runs the whole length of the cochlea because this is what the cells reside in. The tectorial membrane has an inflexible top. So it is there, and some of the hairs sit in the tectorial membrane. So it is inflexible. It cannot move, but the basilar membrane, which runs the base of the cochlea, which is right under here. So this cannot move, but this can move. And notice how these little hair cells are embedded or touching up here the tectorial membrane as well. So these hair cells, these are neurons with mechanoreceptors. These are receptors that mechanically open ion channels by basically physical movement. So they connect between the basilar membrane right here and connect to the tectorial membrane with these stereocilia right here. So these are the things that are connecting those two. And the movement, of this and these interacting together also influences the vestibulocochlear nerve. Right. So how does this work? Looking at a little bit more detail, and let me check my next slide. Okay. So auditory transduction. This takes place at this organ of corti. So here, remember, this is inflexible, but this is semi-flexible down here. So the sound waves, as they come in, cause the fluid in the cochlea to move the basilar membrane up and down because it's flexible. So this is this space right here the sound waves cause this to move up and down. This then causes the stereocilia to move back and forth against the tectorial membrane. So it's can't move up here, they're embedded, but this part can move down here. So it's woohoo. And then you get this movement that is coming from below at this inflexible tectorial membrane. And this would then have to cause the stereocilia to bend or to move because one part is moving and the other part is inflexible. So what happens when they bend or move? Well, these are these mechanical receptors. And so this is this mechanical opening of the potassium channels, which enter the hair cells and then depolarize the membrane and bringing about changing signals and stuff like that. So it's these pieces here, these are some of the outer hair cells. These are some of the inner hair cells right here. They each have slightly different functions, but they are all involved in sound. And yeah, they didn't say anything on here. All right, so go back here. So this is basically how, this is the key piece here, that you have these hair cells, and the hair cells really get the name from the stereocilia because it looks like little hairs sticking up. And when the membrane moves with the hair cells in it, then the stereocilia bend, and that bending basically says hey, we're being moved, we are being affected by sound, and it is occurring at this particular frequency. And then the brain basically takes that, pull all that information into for processing, because the basilar membrane and within the cochlea actually is what's called a totonic map, where I think if I go back here, so this right here, you can see these tones change, and these cumulatively represent the tone of space here. So this is a totonic map where the tones consistently shift, and they are on the basilar membrane, and the change reflects the change actually is specific in terms of how the organ of corti is developed to detect these sounds. All right, So getting back to this. So they had the forgot I had the schematic right here. So this basilar membrane is what's called tonotopic, and that meaning that it is a map of these tones that that you have these spreading down in this organized fashion from high frequency to low frequency. So high is near the oval window and low is further down the membrane. We mentioned this earlier. And higher frequency means shorter wavelengths. So the traveling wave pushes more on the basilar membrane closer to the oval window. Whereas the longer, is longer wavelengths. And so the pressure on the window is different and less frequent. All right. So these hair cells, they correspond to particular frequencies. Then this information, how does this moves from these hair cells? These affect the vestibular cochlear nerve. And then what? So this right here shows aspects of the auditory pathway. So the hair cells affect the vestibular cochlear nerve. Then the nerve affects the cochlear nucleus. So here's you can see here, this is aspects of the nerve. The nerve affects the cochlear nucleus. And then this affects over here with the, oops, where is it? Going to the superior olivary nuclei right here. And then you go to the inferior colliculus and you can see the information from both the right and the left. And then from there, it goes to the medial nucleus, which is right here, again, medial, so it would be towards the inner side of the brain. And then it goes there to the auditory cortex. And so you have this sort of basic, processing up here. And then as you would expect, some fundamental processing occurs sort of along this route, primarily for, particularly for orientation responses. We have preferential processing for things like our name, that we can basically almost sort of like recognize it, and process it sort of especially faster. So some of these types of things are postulated to kind of have this earlier processing that is in these regions. And then it heads to the primary auditory cortex. And here, any sort of, you would further have like a focus on particular auditory information, particular auditory stimuli of importance. So this would be your name. This would be for parents. The cry of a child can be really orienting. The voice of their children. I mean, everybody has some of their own sort of orienting responses, but those types of things would also be processed very early in the auditory cortex. So once we head into the auditory cortex here, you can basically see this by sort of lifting apart, what you've got here, which is the upper lobe, which is heading into the frontal and the parietal, and you have the sort of in the temporal lobe, which is where you're sort of pulling apart that. So you're trying to get at this sulcus right here to pull that apart. And then you can see more where sort of the primary auditory processing region is, which is right in here. And this also follows a map in the sense that this corresponds to the apex of the cochlea, and this corresponds to the base of the cochlea. So even along here, you're going to continue to have this tonotopic map of the auditory cortex that organizes all the information by frequency that maps on to the actual receptor organ, excuse me, the cochlea. All right. So some of the ways that I should say, before I'm trying to think if there's anything I would add before we shift from the auditory pathway. we're going to be heading later into the chapter learning about language. So this is coming into part 2. So we'll be hearing a little bit more about how sound is processed. So we'll be coming back to this information in, in the very near future. And so the information here is really starting to understand sort of, okay, the mechanisms and the pathways of how you are having this sort of the sensory stimuli, whether it's visual, chemical, or the sound waves, that we have particularly well honed and designed receptor neurons, that of course vary by each stimulus. And but the pattern of processing many of them is similar. So looking at, here where the action potentials are the same, but then the types of receptors may have different sort of functions or ways of being activated because they're getting information from the external world. So it's a little bit different than, the cell to cell communication. It builds off of that. The cell to cell communication is like we're all action potentials, but that again, taking that external sort of information and bringing it in, there's going to be at some place where again you have these receptive neurons that interact with the stimuli, and that's where you have that transduction occur. And there's similarities in terms of thinking of how the retina works and how the cochlear membrane works, and even some of the chemical senses in terms of how the information is basically processed. So in terms of just sort of broader understanding, this, this will help also establish a better way to kind of fill in some of the details in terms of how everything is processed. All right, so these are some of a little bit of the fun things that you can have with vision and hearing interactions. So then now that we've covered vision and hearing, the McGurk effect, it's very cool. It is by far best viewed via video. So the textbook provides video, but you could look it up on YouTube, and there are so many of them. And it's one of the few sort of perceptual phenomenon that regardless of how many times you see it or participate in it, it still works. So basically, and you really do have to experience it, but you basically have this sort of mixed up, or they have a bit of a mismatch to show, how this mismatch is to show how the way other people pronounce the words that they are saying, is we use that information to determine, to anticipate what the sound is. So we can actually look then at the way people are pronouncing. So this is what you would do with reading lips. So people can basically, reading lips, they can get better at sort of like, oh, this lip movement corresponds to this aspect of sound production. And so the McGurk effect is basically, okay, you have this lip movement, and we know that these things tend to correspond to certain sound productions. So T, ga, 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 ba, 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 da, da, da. So you have different ways of pronouncing each of those words, which is the stuff that's used in this example. And so if a person is, their lips are saying, as in the example here, ga ga, but the sound from the monitor is coming ba ba, then the person actually will perceive it as an entirely different sound because of those two mismatches. And this can happen all the time, actually, in everyday life, where you can't hear all of what somebody says, and so you use it to fill in the blanks, but there is oftentimes a bit of an assumption there, and sometimes you got the beginning of the word, but you kind of, there's lots of similarities with the beginning, and then with filling in the rest of the word that you didn't hear, So these are the types of things that where we rely on lip reading and we rely on visual information in aspects of our hearing. And so the McGurk effect really just demonstrates that process. And synesthesia is actually a condition where some people have perceptions that cross sensory modalities. So people might talk about hearing words or sounds and seeing colors that represent those words or sounds. There's all different types of it. And so seeing colors and hearing sounds in terms of certain types of words or letters and stuff like that, that's one of the more common representations of it. And so this would be right along this is sort of someone who has synesthesia, they would like 0 would not just be 0, it would be navy blue, and 1 would be silver, and 2 would be yellow. And this is literally how they experience the reading the number and experience the color perceptual sensation at the same time. And so these are sort of here's the primary numbers, and then you can see how 10 would look like this, and 84 and 56. So this is also pretty interesting because for us, when we think of 5 and 6, in terms of 56, is an entirely different number. But for a synesthete, then you would have like the 5 and the 6, because it's a perceptual phenomenon. Then you would get both of those colors with that sort of combined experience, and it's not necessarily like a new color formed between 5 and 6 or something like that. So they have both of them sort of experiencing at the same time, and usually right around the actual particular stimuli. So they would see the stimuli as these colors as well. So this is synesthesia. So both of these are pretty cool. I encourage you to sort of look into them online. There's just some really cool videos. And this, I think, is the end of part 1. And the next for part 2 is actually when we're going to be heading into the neurobiology of language. So I will see you for that soon.